All right, so I think we'll go ahead and get started. Sorry, everyone, the pizza arrived a little late, so we're getting a late start, but everyone's happy with their pizza. So welcome to the Tools and Technology Seminar Series. I think most of you know, so it's just a weekly venue to hear about and have conversations about tools, technologies, methodologies of a variety of types. There's a sign-in sheet. I think most people signed in. If you didn't, please do so. It just justifies us having the pizza and proves that there are people here to it. So that, I will introduce our speaker. Today's speaker is Peter Orchard, who is a graduate student in DCMB in Stephen Parker's lab.
Great, thanks for coming, everyone. So today I'll be talking about ATAC-seq, which is an assay we use a lot in our lab. Probably a lot of you work with it as well. I'll be talking in particular about quality control and about a software developed that. So for those of you who don't do much genomics, I figured I'd first explain why it's important. So this is the world's most naive model of gene regulation. Basically, in each one of your cells, you've got 2 meters of DNA, and it's not just kind of free floating around in the nucleus. It's actually really tightly packed around these proteins called histone proteins. So if your DNA is represented by this kind of string here, it's wrapped up around these blue histone proteins. And the complex of the two is called chromatin. So that's the DNA protein complex. When the DNA is kind of really tightly compacted like this, genes can't really do much. So you can't have the gene being read to produce RNA, just because you can't have any kind of necessary proteins getting to the gene, and so the gene is off. In order to basically turn a gene on and regulate gene expression, you need to have unpacking of the DNA. And so when that happens, the DNA becomes accessible for other non-histone proteins to come and bind. These are called transcription factors, and this is what allows genes to be turned on. So these regions of open chromatin, which are scattered throughout the genome, tend to be important because they're areas of DNA sequence that is important for gene regulation. You generally see them right in front of genes, or you can also see them within genes, or just really scattered throughout the entire genome, even very far from. So because these regions are very important for gene regulation, people would like to know where they are. And there's a couple of high throughput assays to do that. ATAC-seq is one of them. It's kind of the modern preferred method. The general idea is you take your population of input cells. You don't actually need that much, only like 500 cells even will do. Usually people use like 50,000 if they have the material. And you isolate the nuclei from those cells, and you expose the chromatin to this enzyme called TN5. So TN5 is a transposase, which basically means it cuts the DNA. And in particular, it cuts the DNA where the DNA is unprotected by histones. So like in this little cartoon, where you have these red arrows, the DNA is a little bit more accessible. And so you get the TN5 cutting the DNA. And if you have 2 cuts of the DNA relatively close to one another, of course, this results in just kind of a free DNA fragment. And you can sequence these and just kind of align them back to the genome. And so the result is that you end up with these DNA sequencing reads that are concentrated in the regions of the genome that have open chromatin. So to give you an idea of what that looks like, this is an example from our data. It's just kind of a linear view along the genome, showing a gene called MyoD1, which is important to muscle development. And you see the ATAC-seq signal here. You kind of get this big pileup of signal right at the start of the gene, which would indicate the gene promoter is accessible. Something else that's worth mentioning here is that if you look at the fragments of DNA that come out of this assay, you frequently see a very interesting pattern. This is from the original ATAC-seq paper, and you basically see an abundance of short fragments. So that represents cases where you have 2 transposase cuts between nucleosomes. That would be like those two arrows there. Then you sometimes see this mononucleosome peak, and so that represents cases where you get one cut on either side of a nucleosome. So that's like this kind of non-nucleosome DNA fragment example here. So you get enough DNA to wrap around a nucleosome plus a little bit of flanking stuff. That's the fragments in this kind of second bump, and then so on for the kind of third and fourth bump. Those just represent kind of additional nucleosomes being added. Yeah.
Can you go back? So what's the general resolution of this ATAC-seq?
So in terms of like if you do peak calling or something. Yeah, so you do peak calling in ATAC-seq as you would in, for example, ChIP-seq or DNA-seq, which is a very similar assay to ATAC-seq. Usually you're looking, realistically your resolution is going to be like 100 or 200 base pairs or something. Yeah, for peak calling.
I just want to add, if you, some people are looking at like where the cuts happen, like to see like where the transcription factor binds. In that case, you get base pair.
Yeah, so you know like exactly where the enzyme cut just from the sequencing read. You can infer that, but in terms of like actually calling a peak, that's usually going to be lower resolution. That relates a little bit to this next slide. So common use cases for ATAC-seq. Really, it's primarily just used for mapping regions of open chromatin across the genome. That's really most of what I've discussed so far. But there are additional use cases for it. At least I wouldn't probably create an ATAC-seq data set specifically to do this. But if you have an ATAC-seq data set anyway for your cell type of interest, you can use it to map out nucleosome positions. You can use it, as Ricardo was referring to look at transcription factor footprinting. So that's a case where you kind of want the per base pair resolution. You want to know where each cut from the TN5 transposase occurred. The idea being that you can then use that to infer precise regions of transcription factor binding.
Yes. But I guess there will be some like let's say unbound DNA, so that will affect the resolution, right?
Unbound DNA.
Yeah, during the sorry.
Yeah. I mean, so you definitely like get reads mapping all over the genome. You certainly have background, but you know, with peak calling, of course, you're selectively kind of ignoring those regions. And if you do something like transcription factor footprinting, where you're concentrating on per base pair cuts, you're going to tend to focus really on the regions where you have kind of more signal anyway. But you certainly get background, yeah. If anyone is interested in transcription factor footprinting, I recommend they talk to Ricardo. So now that you know how ATAC-seq works, you can think about some of the expected characteristics you would expect a good ATAC-seq data set to show. One that everyone always looks for is just the enrichment of ATAC signal around transcription start sites. So like in that example gene I showed you earlier, many gene promoters will be open, and therefore you expect much of your signal to come from near gene promoters. And so that's usually quantified as transcription start site enrichment. More generally, kind of related, but more generally, you'll do peak calling as you do in ChIP-seq. And you just expect a high fraction of your reads to land in those ATAC-seq peaks. So you expect basically concentration of your ATAC-seq reads, high concentration in particular regions of the genome. People generally look at the fragment length distribution, since you expect that to look very different than just like randomly fragmented DNA. You often see that kind of bi or trimodal distribution, like I showed earlier, that interesting fragment length distribution. But frequently, you just will see like the very short fragment part of the fragment length distribution, and those data sets can also be okay. But you have some expectation for what the fragment length distribution should look like. And so you'll generally check that. Mitochondrial contamination is a common problem in ATAC-seq, and so there are some data sets out there where you're getting like 50 to 80% of your sequencing reads being mitochondrial, and you tend to filter those out bioinformatically. So ideally, you would have low mitochondrial contamination as well. And then really you want to check for all of the same stuff you check for in any other NGS assay. So you want to make sure you're getting good alignment rates. For example, you would be interested in your duplication rate. Really high duplication rate might indicate basically your PCR amplification of the library was kind of too extreme. So stuff like that. So a couple of years ago, this was especially the case, but of course, you want to do QC on your data. You want to look for all those characteristics that I just discussed. And so you'd like some tool to do that. So there are a couple out there. ATAC-QC in particular has been around for a couple of years. This is from the ENCODE consortium, if any of you are familiar with that. And this is kind of a built-in part of their pipeline, so it's not really a separate tool you can install easily. But they give you an HTML report, kind of one report per sample. So that's one tool out there. There are a couple of other ones that are newer. There's at least one that is an R package, so it's kind of you need to use R and you load in your data and you can produce some plots through that. The newest one that I'm aware of was just published this year. This is kind of the nicest of these three, in my opinion. It's just a command line tool. And then you can additionally load some of the output of that tool up into a web viewer. I've generally found it work pretty well. A major shortcoming of all of these is that you're really getting one report per sample. And so anyone who's like run fast QC and had 20 samples is aware of the fact that that means you're looking through suddenly 40 reports or something like that, and it's kind of a pain. Additionally, some of these only work on certain genomes, so like ENCODE's tool only works for human and mouse. So this was a problem that the lab was dealing with a couple of years ago. So we set out to figure out what would you want in a QC tool. And these are some of the characteristics we came up with. You want something that's easy to integrate into any pipeline. You don't want it to be built into some kind of pre-compiled pipeline like ENCODE's is, for example. You obviously like compatibility with any genome. You want to make it easy to compare across samples. So kind of avoiding that one report per sample, because if you've got 150 samples, that becomes kind of a pain. You'd like something that's human readable, but also machine readable, because you'll often be loading in kind of files into downstream analyses. Otherwise, you just like it to be kind of accessible and easy for experimental folks to look at as well. And you'd like things to work on a per read group basis too. This is important because single nuclei ATAC has actually come online now. This is something we're doing in the Parker lab as well. And so being able to run it on a single FAM file with all of your single cell data in there is nice. So we developed a tool to do this. In particular, I was John Hensley, who did the vast majority of the software development. He was a software developer previously in the lab. He's since moved on. But this tool is available on our lab GitHub. So that's the first link there. There's also a demo, which I'll go over in a minute, in case you want to try it out before loading in your own data. So the idea of ataqv is it's kind of as modular as possible, again, so you can fit it into an existing pipeline. So you have for each of your samples a BAM file, which is just kind of a file of aligned reads. It's a standard file you see in genomic analyses. You additionally need to know the organism from which it came, just because there's a little bit of chromosome metadata that is related to that. Different organisms have different chromosome naming schemes. So you need to tell it which organism it's from. Kind of metadata for all of the common organisms is built into the software, but you can also provide metadata for your genome of interest. And then there are a couple of optional files that you would generally want to provide. So you would usually provide a file of ATAC-seq peaks so that you can collect statistics like the fraction of reads and peaks, kind of concentration of reads in those ATAC-seq peaks. You should additionally provide a transcription start site file so that it can calculate things like transcription start site enrichment. Technically, those last two things are optional, but yeah. So you just run it through ataqv. ataqv is a basic command line tool. And the output of that is a human readable text file, but additionally, you get a JSON file. So that's kind of the machine readable component. And then actually, if you have multiple JSON files, because you've run it on multiple samples, you pass it to another part of ataqv, which is this little makearv command. And that combines the JSON files from all of your samples and creates a single HTML viewer. So just to give you an idea of what that looks like, this is the demo from our lab website. So you get a report that looks like this. You have your samples across the top. The naming scheme in this case isn't very interesting, but of course you would give your samples more informative names, I'm sure. So you see the fragment length distributions for each sample. Again, you kind of see that stereotypical ATAC-seq fragment length distribution. You can, there's some scatter plots. You can change the y-axis in particular to look at different alignment metrics. You have this plot of TSS enrichment. So again, I said you would expect the quality ATAC-seq library to have good TSS enrichment, and so you can a curve like this, where what this is doing is you're basically aggregating all of your data across the genome with the transcription start sites at position 0. And it's looking at the signal kind of near the TSS relative to the signal kind of further away from the TSS in the background region. So there's that plot. There's a couple of additional plots, which I won't go into detail on. You also get a bunch of tables. So you get information about the percentage of reads that pass filtering. You again get kind of TSS enrichment statistics. You can sort on all of these tables, so you can quickly see just what's the range of your metric. Information about the total number of reads you have, various mapping statistics in case something with your mapping is funny, duplication information, a little bit of information on for the reads that didn't map, why didn't they map, and a breakdown of basically what percentage of your reads were autosomal or mitochondrial, and for each of those autosomal or mitochondrial reads, a breakdown of what the duplication rate is, because the duplication rate is often very different for the mitochondrial reads than it is for autosomal reads. You get a little bit of information about your peak calls as well. So that demo is available on our lab GitHub in case you want to play around with it yourself. To give you an idea of what it takes to run it, we haven't done any really super formal benchmarking, but when John was putting it together, he did do some basic checks of resource use. So he created one FAM file that was basically an aggregate of all of the data in the report I just showed you. So for those of you who don't work a lot with FAM files, this is a big one, 1.1 billion alignments. Normally for a single library in our lab, we might be looking at like 50 to 100 million alignments. So this is a sizable file. If you don't give it a file of peaks, it can run that in 20 minutes with only 2 gigs of memory. The same file with peaks collects some additional statistics and does some additional computing, so it takes a little bit longer, 40 minutes in this case. Still uses only 2 gigabytes of memory. So basically, you can run this even on a very large file, even on your laptop. Yeah, it's also nice. So as I said, we do single nuclei data in our lab, and so it's nice to have a tool that runs very quickly on all of that data. So that's the tool. I'm frequently asked by people who use it what QC thresholds that I use, and my advice is always, well, take a look at some of the public data that's out there. Take a look at the distribution of your own QC statistics. But basically, it's hard to give really precise values. We found, for example, TSS enrichment can change radically depending on the list of transcription start sites that you use. You also might expect different cell types will show different kind of genome-wide chromatin accessibility patterns. So something like embryonic stem cells might just have more accessibility in general than a more differentiated cell type. And so you might expect different QC metrics from those lines naturally. Similarly, just the purpose of the data should be taken into account. So in general, it's good to look at the distribution of all of your metrics and compare it to public data from similar cell lines. So the ENCODE consortium does put out their own ATAC-seq standards, since they're kind of creating a big public resource. They have very, very set standards for each of the genomes that they use. So you can look there to get a general idea of what you might like. But also, kind of on a related note, we're pushing ataqv through review right now. So we've got a manuscript for it. It's under review. As part of that review, we actually did a survey of all of the public data that we could find for human and mouse. This was back in December 2017, so it's a little bit out of date at this point. But nevertheless, it's got hundreds of libraries in there, including some single cell data. And so if you want to know, kind of compare your data to existing public data, this is a resource for that. It's also, if you're looking for a public data set to use, or you have interest in a paper that does ATAC-seq and you want to kind of just get an idea of what their data looks like in terms of the quality of it, you can also have a look, and it will hopefully be helpful in that respect. Also, as part of that manuscript, we wanted to do a couple of experiments to look at things that we thought might bias ATAC-seq results. So I'm going to run through those pretty quickly. I'll explain why each one of these is interesting in turn. So the first thing that we looked at was the ratio of TN5 to DNA. So as I said, with ATAC-seq, you kind of isolate your nuclei and you expose the chromatin to TN5 enzyme. And one thing that we do in our lab is, of course, you always use the same amount of TN5 enzyme, but we additionally count nuclei when we do this experiment. That's not always done, but we felt it was important just because if you perform nuclear isolation on kind of multiple times, you're not always going to get the same efficiency. And so even though if you're performing some ATAC-seq protocol, you're using the same amount of TN5, the amount of DNA going into your experiment may vary. And so you're kind of changing the ratio of your material to DNA, or your material to the enzyme. And to demonstrate that, we asked a rotation student in the lab to perform a number of nuclear isolations, and we kind of scored the efficiency of each, just based on how many input cells do you have, and then how many nuclei do you get out. And so this is over 10 different nuclear isolations, same cell line, same day, same reagents, presumably. Basically everything the same, but you nevertheless see, in one case, for example, you get as low as 20% efficiency, whereas in others you're getting something like 55% efficiency. So there's a 2 to 3-fold range in your efficiency, which means that the number of nuclei, if you're just counting cells and then you do your nuclear isolation and use that as input for your experiment, the number of nuclei you have going into each one and vary several-fold. And so we were wondering, how much does this matter? How robust are ATAC-seq results to changing the ratio of enzyme to input material? And so for that, we set up this experiment. This was done by a previous postdoc in the lab, Yoshi Kiyono. Some of you probably know him. We started with GM12878 cells, and on one dish or flask or whatever they hold GM12878 cells in, you perform nuclear isolation, and you get you count out then 350,000 nuclei, and then you split those 350,000 nuclei into 7 pools. And with each one of those pools, you perform ATAC-seq with a different concentration of TN5. So that's just, again, changing the ratio of enzyme to DNA. And so you're getting 7 libraries out of each nuclear isolation. And we did this 6 times. So then you have a kind of 6 by 7 or 42 libraries. And we just wanted to see, how does QC change if you do this and some of your downstream analyses. An important note here is, for this one, we held PCR cycles constant because we find that the number of PCR cycles that you would generally do under the protocol can co-varies with the amount of TN5 that you're putting in. But we actually replicated the experiment and many of the findings in a second experiment where we did not hold PCR cycles constant. But that is a caveat for some of them. So you end up seeing actually pretty drastic results in some cases. So this is an example of that. On the left here, I'm showing for one of the replicates the fragment length distribution under 3 different concentrations of TN5. So this black distribution is the fragment length distribution kind of under our normal TN5 concentration. If you reduce the amount of TN5 you add in, you see a shift towards longer fragments in the fragment length distribution, which intuitively makes sense because that enzyme cuts DNA and you're putting less of the enzyme in. Therefore, you expect a lower density of cuts and longer fragments. And indeed, if you double the TN5 instead, you get shorter fragments. So you kind of see the shift in the fragment length distribution that you would naively expect to see. You also see a shift in other metrics as well. So this is for these same 3 libraries, the TSS enrichment. And so you can see that in this case, if you reduce the amount of enzyme you have going in just 2-fold, you see a drop of a couple of points in your TSS enrichment. If you double the amount of TN5, you see a move in the other direction. You get an increase in TSS enrichment. And to show that I'm not cherry picking this, this was highly replicable. This highly replicable across all 42 of our libraries. So there's all 6 replicates, just plotting the TN5 concentration against the median fragment length. So you're getting shorter fragments as you increase the enzyme. And we consistently saw an increase in TSS enrichment as we increased the TN5 as well. The relationships aren't as clean if you allow the PCR cycles to vary. So you especially for fragment length, you see a similar trend, but it's definitely dirtier. Another example of this is just looking at the fraction of reads and peaks, so kind of a quantification of signal to noise in some sense. And you again see a very striking relationship with the amount of enzyme. And this is very, very visually apparent as well. So if any of you are familiar with the UCSC Genome Browser, here's some screenshots from that. So this is just visualizing at 2 different genomic loci. What does the ATAC-seq signal look like? And on the left, it shows a promoter. You kind of see as you increase the amount of TN5 moving up, you clearly see that the signal becomes much stronger. Similarly, on the right, it's the same pattern, kind of further away from a gene, though. If you just quantify where in the genome reads are landing, you again see some interesting patterns. So because people who do ATAC-seq are usually looking for these genomic regulatory elements, these regions of open chromatin, which tend to signify enhancers or promoters. So that's the orange or the red line. We wanted to know, do you see an increased concentration of reads in those regions? And indeed, as you increase the amount of the enzyme, even 5 times over what we usually use, you continue to see an increase of the concentration of reads in these regions. And you get substantially fewer reads in these regions of the genome that one isn't really interested in. And then kind of one last interesting one that we found is as you increase the amount of enzyme you add in, you substantially change the mitochondrial content of your sequencing reads. So in this case, we dropped from 60% at the kind of lowest concentration of the enzyme, and you can get it down to 30% by like maxing out the amount of the enzyme that you use. Yeah, so an important caveat here is, of course, we can only say that those findings hold over the parameter space that we tested. So presumably, if you increase the amount of enzyme you're adding in enough, you basically just obliterate everything, and your results get worse. But in general, in the range that we tested, we saw that increasing the enzyme really shifts the fragment length distribution, and you see kind of an increase in PSS enrichment, fractions of reads and peaks, fractions of reads falling into enhancers and promoters, and a decrease in mitochondrial contamination as well. There are a number of other QC metrics that correlate as well. And so if you have any interest, you can look at the manuscript in review when that comes out. So the second kind of bias that we wanted to take a look at was also related to the fragment length distribution. So if you think that different parts of the fragment length distribution are kind of enriched for different regions of the genome, so you could say promoters, for example, tend to be overrepresented in the very short fragments, and enhancers or other type of genomic elements tend to be relatively enriched in the longer fragments. We were wondering if you kind of subsample from the fragment length distribution, how does that affect your results? And so one way that this occurs in real life is actually related to the way in which DNA is sequenced. So when you perform kind of modern Illumina sequencing, you're taking your DNA fragments, you kind of add on your sequencing adapters, and when they go into the machine, they're binding to this thing called a flow cell. So the DNA kind of sticks to the flow cell with one end. And in the end, when you do your sequencing, you're actually not sequencing just that molecule. You're sequencing a bunch of copies of that molecule, like shown down here on the left. And so as intermediate steps, you add in your DNA, it sticks to the flow cell, and then you do a bunch of PCR amplification, basically within the sequencing machine, in order to get these clusters representing a bunch of copies of the same molecule. And the way that this is done is you have your molecules actually bind over. It's called bridge amplification. The molecules kind of make this bridge, and then you have amplification, and you get another copy, and you just repeat this procedure again and again. Interestingly, you end up with these clusters, but throughout the amplification process, clusters kind of compete with one another for real estate on the flow cell. So like the blue and the green are kind of close to each other in this case. And so, if they're bending over, but they're kind of trying to bend over to the same spot, you end up with these clusters competing with one another for amplification. And that actually introduces a bias because shorter DNA fragments tends to kind of bend over and make that connection easier. So if you have two clusters near each other, then whichever cluster has the shorter DNA fragment will tend to be preferentially amplified. So the end result of that is that if you increase the increase the concentration of DNA going into the sequencing machine, you get an increase in the number of clusters, and you get more competition between clusters, and that results in theory, kind of a shorter fragment length distribution. So we were looking to kind of test this in our data and see, is this effect actually measurable in real life? So we did a second experiment for this. This was very much like the first experiment, actually. But the kind of important point here is that we created 21 libraries, and we sequenced the same exact libraries twice on 2 different sequencing runs. And on one sequencing run, we kind of loaded the DNA kind of at a higher concentration. So we got like 500 million of those little clusters. In a second sequencing run, we again threw in those same exact DNA libraries, but we only ended up with like 400 million of those clusters. So you're changing the density by about 25%. We were wondering, okay, so what effect does this have on the data, if any? And you do actually see an effect. So this is a plot of the median fragment length for each library. Each point is one library. And on the x-axis, it's showing the median fragment length of that library, basically according to the sequencing reads from the low cluster density run. And on the y-axis, it's the same, but from the high sequencing lane cluster density run. And so you see this shift basically off of the diagonal, such that if you take the same library and you sequence it at a lower cluster density, you see a shift in this case of like 10-ish base pairs in the median fragment length. And this actually ends up affecting, basically what this means is you're kind of subsampling the fragment length distribution, right? You have a true fragment length distribution for your ATAC-seq library, but when you sequence it at a really high cluster density, you're preferentially sequencing the left part of that distribution. So you're kind of seeing the library from a slightly different angle. And as a result, you see changes in other QC metrics as well. So this is same as the previous plot, except instead of showing median fragment length, showing TSS enrichment. And so you can see if you sequence at high cluster density, where you're selectively sequencing shorter fragments, the same exact library looks like it has higher TSS enrichment. A shift of, in some cases, like 2 or more points in this case. Yeah, so I think we're just right on timing. So that's kind of a preview of ataqv, including some of the stuff we did for the manuscript. That's currently in review, so hopefully sometime in the next couple of months it will be available. If anyone has particular interest, I imagine it would be okay if we kind of shared with you what we have now as well. So thank you.
Use bioarchive in the lab?
We do. This.
What's the
usually kind of as we're submitting it, unless we have a grant that we really want to have a DOI for. Yeah, but generally, when we submit or shortly before or after, we post it on bioRxiv. This is in review. This has actually not yet been posted, kind of because we've been busy with other things. Yeah.
So does the sequencing part give you like a cluster density guideline?
No, so it's actually something pointed out by one of our reviewers here was like, well, how much does it matter if you see an effect due to sequencing finding cluster density? Because everyone's just trying to maximize the number of reads they get anyway, right? Which means you kind of load your DNA at as high of a concentration as you can. That allows you to just kind of still get reasonable data out. So generally, people aren't going to be thinking about it, but still, it's something that you should kind of be aware of. And you do actually, you can see this effect times in public data. So you'll see the same library being sequenced across multiple flow cells. So kind of having in the SRA system, there's different accessions for if you're running like the same library across different runs, and you can sometimes pick this effect out. But it's nevertheless something that you should be aware of, I think.
Any other questions?
Yeah.
You guys done any comparisons of comparing the fragment distributions you get from sequence data to the like to the bioanalyzer type for sequencing?
So I know that we look at bioanalyzer before. I know you generally, if you have a bad bioanalyzer reading, the results, the ATAC-seq results after sequencing are predictably bad. We haven't done anything really systematic to try to if you attempted to quantify the bioanalyzer fragment length distribution and compare it to the final results out of sequencing, we haven't really done that. But anecdotally, there's certainly some relationship between the two.
Thanks.